untuk melakukan identifikasi DNA atau molekuler dari suatu bahan alam. Jadi kan yang sebelumnya sudah belajar mengenai bagaimana potensi untuk mengolah atau menganalisis potensi bahan alam, cuman ketika kita mengetahui kandidat target dari bahan alam yang ingin kita kaji lebih lanjut, tentunya kita harus mengkonfirmasi identifikasi tingkat identifikasi paksa dari bahan alam tersebut. Nah, kalau secara morfologi mungkin cenderung agak susah ya, sehingga kita tetap harus menggunakan pendekatan molekuler. Untuk hari ini apakah merupakan peserta dari memang kuliah farmasi Bahari atau mungkin ada peserta luar juga? Bagaimana? Oke. Ya, nanti diharapkan kalau bisa on kamera ya, karena kita akan berdiskusi langsung dengan narasumbernya. So, mungkin saya juga akan menggunakan bahasa Inggris dan juga dengan bahasa Indonesia sekalian. Okay, so good afternoon. Today we will start our lecture. The next for today's meeting, we are going to do hands-on practice on DNA molecular identification of natural products. So our tutor today is a senior lecturer from University of Malaya in Malaysia. She is Dr. Poong. So welcome, Dr. Poong. Thank you for joining us today and providing your time to teach about the basic techniques of DNA barcoding. So Dr. Shewan is actually my senior during my time in master studies in University of Malaya, and belongs to Professor Lim Teck Im's lab, which is the Institute of Ocean and Earth Sciences. And well, Dr. Poong has already vast, really deep experience on algal molecular taxonomy, especially algae or macroalgae and seaweed. And not only, she also has diverse research in gene and metabolites expression, studying using omics tools, genetic diversity and population structure of not only seaweeds, but also fish. And there is also her work on analysis or evaluating biochemical stress response and adaptation to climate change, and also shifts in microbiome using metagenomics. So I think her publication, you can already see that she already has a lot of work published in Scopus database, and also this is her resource ID. If you want to reach out to her, you are most welcome. So Shewan, probably you want to greet the audience, and if the audience wants to say something to Shewan, you are most welcome.
Yes. Hi, good afternoon to everyone. I hope you can hear me clear and well. Okay. Before I start, I need to apologize because I'm still nursing a persistent flu, so you will hear me coughing on and off during the lecture. Please excuse me for that. So Dr. Eka, do I start now, or do we need to wait any more? Is it okay for me to start?
Yeah, it's okay for you to start.
Okay.
Can everyone already hear Shewan's voice? Hello? Oh, okay. Yes.
Okay. So throughout the session, if you have any questions, can you just raise your hand, and then I'll try to address the questions. So I'm signing in from two devices so that I can see your questions. You can also type your questions in the chat. I will probably check from time to time if we have a break later on. Okay, so now I'm going to share my screen now. Okay, so I think the screen is up. Okay, thank you to Dr. Eka and the organizers from University Mataram for inviting me. Okay, to give you or probably to share my experience in the bioinformatic workflow for DNA barcoding of marine products. Okay, so this session will be divided into two. Today, I will give you the first half, and then on next Monday, if I'm correct, then we will have the second part of this session because it's quite a long session, right? Okay, so you can call me Dr. Poong. As Dr. Eka explained just now, I am based at the Institute of Ocean and Earth Sciences in University Malaya. So even though the example that I will be using today for this training session is seaweeds, but actually what I'm going to share with you applies to all other marine organisms, okay? It can be corals, it can be seagrasses, fishes, turtles or whatever there is, okay? So you don't have to worry, just go through this with me, all right? Okay, so before we start, I would like to remind everyone that you should have downloaded these programs, which I have shared with the organizer earlier on. Okay, so you should have these 5 programs in your laptops or your computers readily for use. Okay, so this will be the outline of my practical session, right? Today we will do the first 5. Okay, so the first thing is I will share with you some of the tips or factors that you need to consider before selecting which molecular marker to use, okay, for your molecular identification. Okay, then we will look into the quality control of your raw Sanger DNA sequencing data. And then I will show you how to view the raw data, or what we call the electropherogram, using one software that we is known as Chromas Pro. But actually, apart from Chromas Pro, there's a list of programs that you can use, okay, as shown here. Okay, some of them are freeware, some of them needs to be purchased, okay? Some of them are licensed software. So for those that are free of charge, usually they allows you to view your electropherograms, okay? But it cannot do contick assembly. So Chromas Pro that I'm using today, it allows us to do contick assembly, okay? So I think you have downloaded the trial version of Chromas Pro, so it should allow you to do contick assembly, right? So if you don't have access to the paid softwares, you actually don't have to worry. You can, usually what I do is I use Chromas Lite, and then I use BioEdit to do the contick assembly. So it's a bit troublesome, okay? It involves 2 different programs, but if you have Chromas Pro, then you can do this instantly, okay? It's much faster if you have Chromas Pro, but don't worry if you don't have this paid or licensed software, okay? And then we proceed to assemble the contick in Chromas Pro, and I will show you how to do the manual trimming. Okay, you need to trim your DNA sequence that you obtain from the sequencing company, because usually there's noises at the front and also the back. Okay, so we need to trim it, tidy it up so that it's nice enough for us to search against the database, okay, the NCBI or the gene bank, so that it gives us a clue on what is the ID of the sample that you're trying to determine, okay? And then this search against the database, we will do it through nucleotide BLAST or BLAST-N, right? Okay. I don't, is there any questions at this point? Let me see. Okay. Right, so if you have any questions and that you want to ask, you can type it in the chat, right? Okay, so let's carry on. Okay, so imagine someone hands you a sample. Okay, it can be fish tissues or whatever. So here I'm using seaweed samples. Okay, so based on what you can see, the external morphology, I need you to narrow it down to the possible identity of this sample, at least to the family or even better, the genus level. So if I give you something like this, okay, this seaweeds, there's so many seaweeds out there, but I give you something like this. So you should narrow it down to probably it's a Carpophycus or a Eucuma. Or if you cannot point it out down to the genus level, then probably the family level, something in the Soleraceae family. Okay, because if you don't narrow it down, then it is like searching for a needle in the haystack. It will be very difficult for you to select which DNA markers to use for PCR identification. So you should roughly ID it. Okay, how do you roughly ID it? Okay, so you compare it with books, right? You should have reference books, right? And those catalogs on or checklists on whatever algae or whatever fish that you have in your country. Okay, so you should be able to match it. Okay, at least have a sort of uh, a first identification of your sample before we go to molecular identification to verify it. Okay, so as I was telling you, the samples that the example that I'm using is Carpophycus. So excuse me. So later on, we will determine what species of Carpophycus is this sample. Okay, it belongs to the family Soleraceae. All right. Okay. So just now I have narrowed it down to it could be either a species of Carpophycus or Cuma. Now next, what do we do? Okay, you can search for available literature. Okay, search for publications like the one I've shown here on the right hand side. Okay, this is a paper on the genetic diversity of Carpophycus and Eucuma. All right, so you go to the materials and methods or methodology section, and then you will see that here it will show you which markers did the authors use. So here they use the COX1 and COX-23 spacer genetic markers. So you have two options here. So it really depends on you. If you have more resources in your lab, okay, then you can do both markers. If you just want to have a quick ID, then probably one marker is sufficient. Okay. Apart from searching for available literatures on what people have used, I would also suggest you to search for the gene bank database. Okay, I will show you this next week, but let me tell you briefly about this. So usually I will ask my students or the participants to search the gene bank database, because if you have your DNA sequences and you want to search against the DNA database, first you need to know what is available in the database, or else you'll be searching A to something that is unknown, right? So you can go to the gene bank database, the NCBI database, and then you search for, let's say I type in Carpophycus. So I wanted to know which are the markers that people usually use and have deposited into the gene bank, so that later on, what I have sequenced out, I can actually use it to search against the database. If let's say you use um, an 18S marker, but there's no 18S marker sequence of Carpophycus in the gene bank, then you can't do any search, right? Your search will return nothing. If there's nothing, if there's no 18S sequence in the gene bank for Carpophycus. So that's what I'm trying to say, okay? All right. Okay, next is I wanted to tell you about primers, all right? So I assume most of you here have some basic knowledge on molecular analysis. Okay, so I think you should know what are primers, right? So primers, they usually come in pairs. You use primers during your PCR amplification. Okay, so example that we are using for the sequences that I have shared with you, okay, is the COX1 gene. primer 43F, which is paired with 880R, and the second set is 622F, which is paired with 1549R. Okay, so note that this COX-1 gene, okay, actually the reason why I chose COX-1 gene is apart from its being used in literature, okay, and also available in gene bank, it's because the topic for today is DNA barcoding, and you should know that COX-1 is a very commonly used DNA barcoding marker, not just for seaweeds, but also for other organisms, okay? Well, maybe not fungus. Fungus, we usually use ITS, but apart from that, most organisms, we use COX-1 as the barcoding marker, okay? Okay, so let's carry on. So the numbers here in your primer name, they usually indicate the position of the nucleotide. Okay, what do I mean by this? Okay, if you look at the diagram over here, okay, so imagine this is your DNA, okay, and then this region, the white color region, is your COX-1 gene, right? So I'm going to use two sets of primers, 43F and 880R, to amplify this region, okay? And then I'm going to use 622F and 1549R to amplify this region. Okay, so these numbers actually refer to the position of the nucleotide within the COX-1 gene. Okay, so you see it's in ascending order. Now, some of you might ask me, why do I need to use two sets of primers? Can't I just use one set, 43F and 1549, then I can get the full line sequence? Okay, the answer is yes, actually you can do that, but it is not as easy as it sounds. Most of the time, it's quite difficult to amplify and also sequence something that is more than 1 kb or 1000 base pair. Okay, so but by all means, go ahead, try to do 43F and 1549R. If you get this amplified band at one go, then congratulations, this is very good. Okay, but if not, then don't worry. You can try to do these two sets of primers. You sequence them. Then after that, we will do contact assemblies to get the full length COX-1 sequence, which is more than 1,400 base pair. Okay. I think yes, that's what I wanted to say. All right. So for this training, we will be using COX-1 sequences of a sample of Kappa ficus. So we're going to find out what species is that. Okay. So I have shared, excuse me, I have shared some DNA sequences with you through the organizer, I think a few days ago. So there's OMD 13 and OMD 20 inside the folder. Okay. So we'll be using that for our practical session today. Okay, next, right. Okay, so I think you can recall last week during Prof. Lim's lecture, her final slide shows the workflow of you started off with DNA extraction, right? And then you do your PCR amplification, your gel electrophoresis, and then you send the amplified PCR bands for DNA sequencing. Okay, so you can either send it out for sequencing to the companies, or you can also sequence it in your lab if you have a sequencing machine. So what is important is the results that you get back. Okay, so what I'm showing you on the screen is an example that I get directly from the sequencing company. Okay, so these are my own the sequences of my own samples, okay? And then you can see here, there's usually a control sequence that is given, okay? So this control sequence is meant for you to check, okay? So this is to show you that if your own DNA sequence is very noisy, not good, then you can check it against the control. So if the control is showing very nice peaks, then it means that there's nothing wrong with the sequencing run. So most likely the problem with the noisy sequence in your own DNA sequence is due to your sample contamination or whatsoever, okay? So the control is meant for you to check whether there is something wrong with the sequencing run or not. Okay, from the next slide onwards, I need you to do this with me step by step, okay? So first thing, I want you to open the folder. Okay, let me share my folder. Hang on. Let me just share. Just a moment. Let me see. Okay. So you should have a folder like this, right? I have shared these sequences with you. Okay. Okay. I hope everyone is following me. If not, then please raise your hand. Okay, I don't see anyone raising their hands. Okay. Right. So in this folder, you can see that there's two types of results. Okay. This one is the AB1. Okay. Another one is the SEQ, the seq file. So there's two types of format that will be sent to you by the sequencing company, or it will be generated by the sequencing machine. Okay. So the AB1, right? The AB1 the AB1 files are actually the raw DNA files where you will see the electropherograms. So you will see the different peaks, okay? And also the DNA sequence, so AGC, GT, those bases. Okay, so this is the raw data that is generated by the sequencing machine. Another one is the seq file. Okay, this is actually only the DNA sequence. You don't see the peaks, the electropherograms. So for the seq file, you can actually use any text editor programs like your Microsoft Word or your notepads, WordPads. Then you can actually view the sequence in FASTA format. Okay, so what we will be using more often is the AB1 file. Okay, so now I want you to open your Chromas Pro. Okay, can everyone please do this together with me? Let's see.
Ada ngikutin nggak ya step-stepnya, Dek? Bagaimana? Atau kecepatan? Halo?
Halo Pak.
Nggak ada ya? Stepnya ngikutin?
Ngikutin, tapi ini ada kendala sedikit.
Do you need me
What's trouble?
to repeat? Apa masalah dia? What is the problem? You're stuck at which step? I can do this slowly, but you need to let me know at which step you have a problem. So you open your Chromas Pro, then you will be able to see something like this. Is everyone okay?
About the others, yang lain udah masalah?
If you have, just raise your hand, then
Yeah, okay. Probably just type in the chat kalau ada if there is any problem.
Yeah, okay. So if there's no problem, then I will proceed, right? Okay, so you open your Chromas Pro and then click File, click New, and then go to Sequencing Project. Okay. So after you have done that, okay, you will see the screen showing something like this. Okay, then we click Add Files. Okay, I hope everyone is following me. Okay, so after you click Add Files, then go to the folder, okay, where you have your sequences, and then select those AB1 files, okay? There should be 4 AB1 files under OMD13. And if you can see here, right, they are actually individual sequence using different primers, 43F, 880R, 622F, and 1549R that I was talking about just now, okay? Select all these 4 sequences, then you click Open, then it should be appearing on your screen like this. Okay. All right, since there's no questions, okay, let's carry on. Now, you have these 4 sequences. So I want to merge them into a single COX-1 sequence. So what we do is, it's very simple. If you use Chromas Pro, you just click Assemble All. Okay. Click Assemble All. All right, then it will give you an assemble contig. Okay, so this contig is 1544 base pair in length. Excuse me. So if you see, before we assemble, each of it is 800, 800, 900 base pairs, right? So after we merge, it becomes 1005 because you remember there's an overlapping region just now, which I showed you between the 2 primary regions that we sequence. So once you merge them, you will get this contig, okay? So if you double click on the contig, you will see your electropherogram, okay? So this is the peaks that I was talking about, all right? So this is the peaks, the electropherogram, and this is the DNA nucleotides, right? Okay, no questions in the chat, then I will carry on. All right, so if you see here, and also here, okay, let's look at this. Your screen, right? You see here, I am scrolled to the far left. Okay, so this is the left-hand side of my DNA sequence. So you will see that it starts with 622F and then 1549R. But if you look at this one, my 43F and 880R should be at the left-hand side. So I expect to see 43F and 880R at this site. And then when I scroll to the right, I should see 62F and 1549R. So what I do is I click this reverse complement, okay? This icon next to the telescope, right? So if you click reverse complement, right, it will show you 880 and 43F over here. Okay, so the reason we want to reverse complement is because we want to get the correct orientation data, especially when you do multiple sequence alignment. If your sequence is in a different direction from the other sequences in the gene bank, then you won't be able to assemble, to align your sequence with the rest of the reference sequence. So you need to get the orientation correct. This is important. Okay, so now that we already get 880 and 43 at this site, okay, now what do we do next? We will, okay, getting the correct sequence is correct. Okay, so you might ask me, doctor, why it starts with 880R and not 43F? Okay, why is 880 on top? Shouldn't it be 43 then 880 in that order? Okay, I want to explain to you by using this diagram. So when you want to PCR, okay, amplify this portion, so your primer will bind here. Okay, and then the DNA elongation will be in this direction. Okay, it's always 5' to 3'. Remember your theory in DNA replication. Okay, try to recall that. Okay, and then on the other strand of the DNA, you have your double helix, right? So you have one strand here and the other strand here of your DNA. So the 880R primer, which binds here, okay, it will elongate in this direction, all right? So at the end of the day, this one will be slightly longer. So you will, which is why when we read it like this, right? So it will start with 880 first and then 43F. So 43F will be having something jutting out here, okay? So that is why 880, 43, 1549R, and then 622. This is the usual condition that you will see, but don't be surprised if you see it starting with 43F first, because sometimes the sequence for your 880R, it might be prematurely terminated during the sequencing. So something might happen during the sequencing run. So this one is a bit shorter. So let's say it stopped here. So when you look at it in an overall picture, you will see 43F first, then only 880R. But otherwise, conventionally, you will see 880R first, 43F, 1542, and 622. Okay, just to give you an explanation on that. Okay. All right. Now that we have already assembled the contig, okay, we already get a single DNA sequence out of 4 that is merged together. What we do is we need to trim the 5' end and also the 3' end. So the both ends, right, they usually includes the primer sequence. So we don't really need the primer sequence because this will affect our alignment later, okay? Okay, so how, before you do the trimming, you need to know what is your primer sequence first, right? You need to know what it is before you can remove this primer sequence. So usually you can get this from literature, right? Like just now you were referring to the paper on genetic diversity, right? So it will basically tell you the primer sequence, okay? So for the forward primers, you don't need to do anything. But for the reverse primers, you need to reverse complement them first. All right. Now, I want you to do one exercise. I want you to list out the reverse complement sequence of primer 1549R. Excuse me. So try to reverse complement this. I want you to do it right now because we're going to need this in the next few slides. So I'm going to give you 1 minute. Can you try to do this reverse complement? I hope you know what that means, right? So if it's AGGCA, then when you reverse complement it, it should be A becomes T, G becomes C. So try to reverse complement this. And then I will continue with the next slide. So for N, you just use by N, okay? N means it can be A, C, G, or T, right? So yeah, I'll give you a minute, right? Is everyone able to follow until now? I don't see any questions in the chat. There's no hands being raised. I hope everyone follows at this point.
Apakah sudah jelas yang diminta ya? Jadi disuruh melakukan reverse complement yang ini ya, yang sequence yang warna merah itu. Silakan. Jika ada selesai langsung disampaikan aja.
Okay, all right. So assuming that you're in the process of doing that, okay, you can carry on. Okay, so let me proceed. Okay, so this slide is just to show you some of the ambiguity code symbols, all right? So if you're not sure whether the nucleotide is A or G, then we use R. Likewise, if you're not sure whether it's C or T, then we use Y. Okay. You can actually get this when by searching for it in Google. Okay, so don't worry. You can find it very easily. Okay, you might ask me, why do I need this? Okay. Okay, you have your sequence, right? If you have 880 and then you have 43F. So one at the top gives you A, and then the other one gives you a G. So what's the decision? Is it A or G? If you're not sure at that point, because both of the peaks are very nice, then you can't be sure which one is correct. So you just put an R there for the time being. Later, after we do multiple sequence alignment with the reference sequences, and then you can see the other sequences are all showing A, but yours is R, then most likely it is actually an A rather than a G. Okay, so you might need to use this sometimes if your sequence is not very nice. Okay, so okay, next. Okay, still no question. All right, now I'm going to show you how you should trim your sequences. Okay, so first we trim the 5' end. Okay, my question is, do you see the sequence of 43F primer in this part of your sequence? Make sure you scroll your cursor to the far left end, okay, which is the 5' end. Okay, so do you see this sequence TCAA inside here? Do you see it? So there's your primer sequence. Okay, so I'm going to remove everything from this point onwards. I don't want all this. And by the way, if you notice, you see here, the peak is not very nice. Okay, it's starting to get nicer towards the middle. Okay, so this one, we remove it. Okay, just delete it. Right, and then we scroll to the middle. Okay, scroll a little bit to the right. Now, what is meant by reliable base call versus noise? Okay. Okay, so if you see there's black boxes here, right? So these black boxes means you should pay a bit more attention, okay, on this position. So the T here is highlighted, okay, because 880R is telling us it's T, okay, with a nice peak, okay, and then 43F is telling us there's nothing there, okay, and the peak is not nice, okay, it's jumbled together. So that is why it's highlighting T. So here you need to decide whether it's a T or something else. So the answer is actually yes, it's a T, because usually at the start of each sequence, you can see that it's not very reliable. So this is what we meant by noise and unreliable base call. Whereas this one, okay, because we already deleted the front part of 880R, and then we scrolled to the right already. So this is starting to get nicer. So these are considered very reliable base call. You can also see the boxes here, right, on top of the T. So the red color box is full. If it's full means it has higher reliability. If it's just half filled, then it has lower reliability. So that is what we meant by reliable base call versus noise. So we don't want the noise, we want this reliable base call. Okay, so I talked to you about T. So C is a similar case. Okay, but this time around, for 880R, it's showing nothing. So this green color box, nothing. And then 843F, it's showing you a C. But bear in mind, here the peak is very noisy. So if I ask you to decide, would you retain this C or would you remove this C? Okay, if you ask me, I would remove it, okay? Because here is much more reliable than here. So if you see here, the boxes are either half filled or very minimally filled. So I don't trust this, but I trust this. So I would actually delete the C. Okay, what about this A? Same case, I would delete it, okay, because there's no A over here. But this A is coming from the noise from the sequencing. All right. Okay. So I will also delete the T and the T over here. So the only thing that I will not delete over here is the first T. Okay, so black boxes indicate that you need to check one by one, but not necessarily need to delete. So at the end of the day, I only delete this, okay, but not the first T. So you need to check. You have to check between 880R and 43F. Which one is the reliable one? Which one is not the reliable one? By looking at the peaks, and also you can see the boxes here. If it's completely filled, then the reliability is higher. So as you see here, I retain the T and then I delete the rest of it. Okay. Now I scroll to even further right. Okay. So there's some more black boxes that is being highlighted. Okay. So this one, let's have a look at this one. Excuse me. So if you scroll it to the right, you start to see that excuse me, 1549R and 622F is also turning up here. Okay, so you will be able to see 4 because this is the part where it's overlapping region. Okay, so here 880 and 43F is showing nothing at this position, and then there's an A from 1549R. So remember I was telling you just now, check the base call reliability. So obviously this is not very reliable. So I'm going to delete this, okay? What about this? Okay, so this is the C, and T. So is it a C or a T? So just now I was telling you, if you're not sure whether it's a C or a T, then you can put Y, right? But if you have actually gained some experience after doing many rounds of this, experience will tell you that this is a C. Okay, so even though this has a very nice peak, I suspect that this is actually, because it's at the front of the sequence, so it might be part of the primer sequence. So I usually won't take this into consideration, even if it's showing in a nice peak. Okay, so I will trust this much more. So I will retain it as C. Okay, same goes with this. Okay, A, right? C. Okay, this one is showing N because there's nothing from the 622F. It shows an N. Okay, so I will still go for C. So for exercises like this, you need to really do this a lot. Then with experience, you will be more confident of editing the sequence. Okay. So just now I have edited the 5' end, and then I move on to the middle, and now I scroll to the back. Okay, so this is the 3' end. So 3' end, you remember just now I asked you to do the reverse complement of the 1549R primer. The reason I asked you to do that is because I want you to see whether you can detect that sequence at the end here, because I want to delete the primer sequence. So if you have correctly done the reverse complement. Okay, so you should be able to see the sequence. Okay, so the sequence is actually TATCA. It's over here. Okay, okay, I'm not sure why I didn't highlight this, but anyway, so this part I will delete it. Okay, so after I delete the primer sequence, okay, I am left with 1400 something base pair out of the 1005. Okay, because I have trimmed away the 5' end and also the 3' end, which consists of the primer sequences. Okay, now that after you trim both ends, okay, so it's already tidy, very nice. You need to save, okay, so you go to File, click Save As, right? I want to save this project so that I can view the electrophotogram anytime I want. So sometimes you might do the wrong editing at this stage, and later you can come back to see the file to double check. Okay, so I will ask you to save in two different formats. One, we click Save As. Okay, then you this one, the Save As type, you click project file, the .gpj format. Okay, the file name, it's up to you. Okay, like in my case, I put it as OMD 13 COX1, and this is after edit. Okay, so the GPJ file will allow us to save this project so that we can come back and double check it later. Now I also want the FASTA sequence, which is the plain nucleotides, ATGC or whatever it is, without the peaks. Okay, it's just a sequence in FASTA format. So you click File, Export to Editor. Okay, then it will come out something like this. Click Save As. All right, and then the Save As type, you choose the FASTA format. All right. Okay, so after you save, inside your folder, you will see your GPJ file and also your FASTA file. Okay, I don't see any questions in the chat, so I assume everyone is following me. Anyone wants to stop me at this point? Okay, there's one question. Oh, okay. You have a problem with your laptop. Okay. All right. He already input the data in Chromas Pro. All right. Okay. So I hope you'll be able to follow me. But anyway, because this session is recorded, and I suppose the organizer will share the recording with you, and I will be happy to share my slides as well. So yeah, should be okay. You missed the next step. So after you input the data in Chromas Pro. Okay, it's okay. Let me finish this, and then I'm going to repeat I will repeat it after this. Okay, so that it doesn't cut the flow. Okay, so inside your folder where you have your sequences, if you followed my steps correctly until now, you should have this GPJ file and then the FASTA file. So if you click open your FASTA file, you will see something like this. It will show you config 0. So I want you to rename this, okay, as OMD 30 COX1. So this one will be this the final sequence that I will use to search against the NCBI database. Okay. Okay. So since there's a question from the chat, let me just quickly repeat what I was telling you just now. Okay, so I think our friend here Okay, so you have already input the data in Chromas Pro. Okay, so I think it's this step, right? So you have this in, all right? Click Assemble All, okay, to merge it into one single config. Double click. I'm going to go a bit faster, right? Because we have actually go through this. So scroll to the left hand side, right? If you see it starts with 622 and 1549, click the reverse complement, this one, okay, so that it will show us 880 and 43F over here. Okay, make sure your cursor is scrolled to the far left, okay? Because if you scroll to the middle or to the end, then it will show different thing, okay? So what I'm asking you to do is scroll to the left first, then only you click reverse complement, all right? Okay, this one I can skip. Yep, skip. Okay, so after we do the reverse complement, then I need you to trim the 5' end. Okay, so my cursor is still scrolled to the far left of the sequence, right? So I want to trim off the primer sequence. So 43F, this is the sequence that you can see from literature. So you ID it, it's over here. So I'm going to delete this. Okay, so just highlight everything from the beginning until here, then you can delete it. Okay, and then those black boxes, right, are those that you need to take note of. Okay, so for instance, the T here, it's being highlighted because this sequence is telling you it's a T, and then this sequence is telling you that there's nothing there, so it's very noisy. So that's why I will retain the T, because this is the reliable one. Whereas for the others, okay, there's nothing over here, but it's very noisy sequence at the bottom. So I'm going to delete this 4. Okay, right? So yeah, delete. I'm going to delete those 4, and then I will carry on the same process from the left hand side until the right hand side. So you continue to scroll, and scroll, and if you see the black boxes, you need to decide whether to retain or to remove them or to edit them. Okay. All right. So this is in the middle. So I'm going to delete this, and then I'm going to retain all this because it's showing reliable base calls. Okay. And then the same thing for the 3' end. Look for the 1549 primer sequence, but for this one, remember it's a reverse primer, so you need to reverse complement it first, which is what I asked you to do just now. Okay, search for it, and then this is the sequence. Delete it. Okay, then we save our config as GPJ format. Okay, right, and also another one. Click File, Export to Editor to save the sequence in FASTA format. All right. So yeah, and then once you have your GPJ file and your FASTA file, click open your FASTA file and rename it. Okay, rename the config 0 into your sample name. Okay, so for our case, I use OMD 13 COX1. Okay, any more questions in the chat? No. Okay, no questions means I assume you are able to follow me. All right. Okay, so let's carry on. Okay, for this practical session, I gave you two sets of sequence. OMD 13, which is the nice ones, and then another one is the OMD 20. Okay, so the reason is because I want you to see what are nice sequence and what are noisy sequence. Okay, so I want you to repeat the steps from opening Chromas Pro, loading the OMD 20 sequences into the Chromas Pro. Okay, assemble it. Okay, double click the config, and then you will see something like this. Okay, can you please do that right now? Okay, let me do this together with you. Okay, hold on. Let me look for my chromast. Okay. Share screen. Okay, my chromast. Okay, so I have my Chromas Pro open. New sequencing project. Okay. Click Add Files. All right. And then go to OMD 20. Load the 4 AB1 sequences. Click Open. All right. So you see all the OMD 20, 43F, 880R, 622F, 1549R, all these sequences inside here. Okay. Now click Assemble. And then I double click this to see what's inside. Now, okay, you can actually play with this, right? If you want to enlarge it. Okay, Y is for you to make the peak higher or lower. Okay, so usually I don't play with the Y axis, but you can do this with the X axis. Okay, so my cursor is at the far left. It's showing 622F1549R. So I was telling you that you should reverse complement it. Okay. So once I click reverse complement, it shows 1549, but you see the cursor is at the right hand side. So we pull it back to the front, then you will see 880R and 43F. Okay. So as you scroll to the middle, when you reach the overlapping region, you will see all four here. Okay. And then when you scroll again to the right hand side, you will start to see only the 1549R and 622F, which is the back portion. Okay, the second part of your COX1 gene. Okay, this is the 3' end of the COX1 gene. Okay. All right. So why did I say this one is an example of a noisy sequence? You see here? Can you see this? You see you have overlapping peaks that are, you know, this, the C and the T. Okay, sometimes you will see something like this. Okay, so this is an example of a poor quality DNA sequence. Okay, why do we have a sequence like this? There's a number of reasons. Okay, your DNA sample might be contaminated. Okay, it could be during your sample preparation, you accidentally mix tissues from 2 different species. Okay, so when you extract, then you get 2 different types of DNA. And then when you amplify the PCR, you get 2 different DNA mixed inside the sample that was sent for sequencing. Another reason is maybe your primers are contaminated. Okay, so don't ask me how you contaminate it. There's 1,000 over reasons how you contaminate it. I also don't know. Sometimes my students also contaminate their primers. Okay, so what you can do to rectify the problem is first we go back one step. So from your DNA sequence, you go back to your PCR amplification. Try to you can either order your new primers if your stock is still clean, it's not contaminated. Because usually when we order the primer stocks, the best practice is to aliquot the primers, okay, into individual tubes. So you use the aliquots, okay. So if the aliquot is contaminated, then you are quite lucky. You can just throw away the aliquots of your primers and use new aliquots from the clean primer stock. But if your primer stock is contaminated, then you don't have much choice but to order new primers. Okay, you need to reorder new primers. So after you sorted out the primer problem, you did your PCR and you send it for sequencing, it's still giving you this problem of noisy sequence. Then you need to go back further one more step, which is your DNA extraction. So your DNA might be contaminated. If that's the case, you need to repeat your DNA extraction process. And this time you need to be more careful so that you don't contaminate it with other DNA. Okay. All right. So this, I just wanted to show you what is an example of a bad DNA sequence. Okay. So you see here, why it is showing K, because here it's telling us it is G and then this is T. So this is ambiguous. That's why Chromas Pro is indicating it as K. But obviously none of this is reliable. Okay. Let me see if I can find. Yeah, so this is also, one is telling us it's A, another one is C, that's why it's showing M. This one is also, one of it is showing T, one of it is showing C. So this is the where you use the ambiguity ambiguity codes. Okay, but frankly speaking, if your sequence come back like this, I will suggest you please don't use this. Repeat your PCR. If it still comes out noisy, then repeat your not to say repeat, you redo your DNA extraction, okay? Or else you will keep getting noisy sequences like this, all right? Okay, so let's stop sharing this and go back to my slides. Any questions? Nope, no questions. Okay, good. Share screen. Okay, so let's see. Okay, so we have come to the final part for today's session. This is DNA, the database search against, which means just now you have your full length DNA sequence that you have already trimmed. Okay, so it's very nice right now. So I want to know the identity of this DNA sequence. So what I do is I need to search this DNA sequence against the gene bank database, okay, or NCBI database. Okay, so because you are searching a DNA sequence, so usually we use NCBI, nucleotide BLAST. Okay, if you have an amino acid sequence, then you can use BLASTX or protein BLAST. Okay, so it depends on your input file. If you're using DNA, then you use BLASTN. If you're doing protein, you can also search against the EBI database, right? Okay, so go to your browser. Okay, do this together with me, please. Open your browser and then you type nucleotide BLAST or BLASTN and see whether you can get to this page. Okay, let me do this together with you. Hold on. Okay, let's see. So does everyone manage to get to this page, the BLASTN page? Yeah, you just need to type BLASTN. Then it will get you to the page. All right, so it's quite simple. All right, so at this page, okay, you see it has BLASTN, BLASTP, BLASTX, all different kinds of BLAST. So we're using BLASTN for BLAST nucleotide. Okay, so just now, you have your sequence in FASTA format, right? Remember the one that we renamed from CONTIG 0 to OMD 13 COX1? So, let's go to your folder. Okay. Oh, do I have it here? Okay, wait, hang on. OMD 13. Okay, so I have my OMD 13 and then my sequence file. Oh, sorry, I think I didn't save this into FASTA format. Oh, sorry, I think I didn't save this. Never mind. Format. Save as. Okay, I'm going to save this in. Okay, right. Open with that. Okay, so copy your FASTA sequence, okay, and then paste it onto the the BLAST page. BLAST. Okay, I'm going to paste it here, right? OMD 13 Cox 1. Job title, if you just click here, it will show you the name from, it will auto detect from your sequence. Okay, the database, we will use standard database, okay? Use nucleotide collection, okay, which is the default. Organism, you can leave it blank. The rest, you can leave it blank. And then for program selection, we always go for the first one, highly similar sequences. Unless it doesn't return any hits, then we will try with the less similar ones. Okay, so always try with the first one first. Okay, so click BLAST. Okay, so it is searching against the NCBI database. Right, so the result came out. So you see your job title is OMD 13. The sequence length is 1479. Okay, so what is important is this one. Okay, so you can see here, it shows Kappa ficus malaysianus, Kappa ficus sp1, alvarezii. So as you scroll down, you will see a long-lived, this is striatus. Okay, so in other words, different species of Kappa ficus are being shown here. So you might ask, Dr. Poong, so which one is the correct one? Okay, how do we determine? You look at the percent identity. So these are the scores, query cover, this is the e-value. Okay, what is important is you look at the percent identity. The higher the percent identity means the higher the likelihood this is the correct ID. So 99.93 is quite good enough, if you ask me. All right, so if you scroll down, the percentage will get lower. So it is 95.71% that this is Kappa ficus alvarezii. So obviously I will go for Malaysianus. Okay. So if you ask me why there's so many Malaysianus here, because each of this is a sequence of different specimen that is uploaded by authors from their publications. Okay. Is everything clear at this point? Let me go back to my slides. Okay. Okay, so if you look at your browser just now, we have the query sequence. Query sequence means what we put in, okay, the input file. Okay, so your sequence is the query sequence. Match sequence is what is managed to be found by the database search. It's also called a heat. So this is, there's also a column for scores. But usually we don't really look at the scores or even the expectation or e-values, okay? What is important is the sequence identity or similarity, okay? So that is the end of part 1. I have also prepared an assignment for you. Hold on. So I think I sent you a file on assignment. So the first one is the exercise that I asked you to do just now. I want you to reverse complement the primer sequence of 1549R. Okay. And then scroll to the end of the sample config, highlight the reverse. Okay, I want you to do this. Okay. First, you give me the sequence, and then I want you to print screen this thing. Okay. And then the second exercise is to trim the config sequence and paste the sequence here. Okay. So for today, you do question 1 and 2, and then probably the organizer can collect the answer scripts and send to me so that I can check it. Okay, so that I know whether you follow me correctly or not. And then the third part, we will do this next week. Okay. So I think that's all for today. Do you have any questions for me? So next Monday, we will carry on with the how to search for reference sequence, multiple sequence alignment, generating a phylogenetic tree. So this tree is important if you want to have more conclusive identification. So if you just want to know the identity of your specimen, you can just do up to step 5. But for publication purposes, usually you will need to show more concrete proof by running a phylogenetic tree. So I will show you how to do this next week. I will also show you how to estimate a pairwise distance between 2 species, okay? And also how you deposit sequences into gene bank. So I think these are some of the things that it's important for students or also researchers with regards to your publication. Okay, so these are some of the things that I hope you will find useful. Okay, so we will cover this 5 in the next lecture. Okay. Any questions? If not, I hand it over to Dr. Eka or the organizer.
Thank you very much, Shewan. I think there will probably be questions along the way when they will start doing it because I'm not sure if most of the participants are they probably have something another subject going on. So I think they can or later view your recording and we will transfer if there are any questions to you. So
Yes,
thank you very
sure.
much for your lecture today. So we are expecting the second part uh, next week.
Okay.
So
Yeah, no problem.
okay.
All right. Thank you, everyone. I hope you enjoyed the session. If you have any questions, you can email me, right? Okay, so I'll see you next week then.
Okay. Thank you very much.
Okay, bye-bye.
Okay.